Okay, now we're going to look at another type of transport called active transport. So with passive transport, what was driving passive transport was diffusion. With active transport, we're going against diffusion, right? With active transport, we're moving materials against their concentration gradient, right? You don't want the ball to roll down the hill, you want the ball to roll up the hill, right? You're going against physics, right? You're going against the physical environment, and that's going to take energy. Active transport requires energy, right? And this is usually supplied in the form of ATP. Active transport also requires proteins in the cell membrane to help the process. So this is always a carrier-mediated process. There are two main types of active transport. The first type is called active transport. Don't shoot the messenger, I didn't get the name of this stuff. Okay. The second type is called secondary active transport. To make your life easier, we're not going to talk about secondary active transport. We're just going to talk about the first type, primary active transport, which is really just referred to as active transport. The difference between the two, by the way, is how the ATP energy is used. If the ATP energy is used directly, that's the first type. If the ATP energy is used indirectly, that's the secondary active transport. All right, so with primary, if you will, active transport, what we're doing is we're moving things against their concentration gradient. And we're directly using ATP energy in the process. So one example of this is an ion pump. In the picture here, we see a proton pump. This is inside the cell, this is outside the cell. You can see there's lots of protons already outside the cell, right? So according to diffusion, the protons want to come in, right? But protons mean acid, you don't want all that acid inside the cell, so we want to pump more protons out. So you want to move these protons against their concentration gradient. Well, if you want to defy physics, you have to be prepared to spend energy. So we're going to use ATP energy, which we see here in yellow, to pump the protons out. Right? Another example of active transport that's really important is the sodium-potassium exchange pump. So this is the sodium-potassium exchange pump. As the name suggests, it's going to move both sodium and potassium. Okay? It happens to move 3 sodiums out for every 2 potassiums that come in. So the sodium and potassium are moving in opposite directions, hence the word exchange. But they're both moving against their concentration gradient, so this is going to require ATP energy. So you can see there's lots of sodium outside, right? But we're going to put even more sodium outside, so we need ATP for that. The phosphate breaks off of ATP, and the attachment of the phosphate causes a shape change in this carrier. And so now the carrier is open on the outside. So now the sodium is going to be released by the carrier and then will enter into the external environment. Once the sodium is released, it causes another shape change in the carrier that makes the carrier more receptive to potassium. So now potassium can come in and bind. And when the potassium binds, that causes another shape change. Phosphate's released, and now the carrier is open on the inside, and then the potassium will be released to the inside. So basically this protein that is the pump constantly changes shape, allowing us to move sodium and potassium against the concentration gradient. What's helping to change the shape of the protein is the ATP, the attachment of the phosphate group and that sort of thing. So that was the sodium-potassium exchange pump. Sometimes people just call it the sodium-potassium pump. For the most part, this pump is going to be moving sodium out of the cell and potassium into the cell, though there are a few exceptions. The ratio is not 1-to-1. The ratio is 3 sodiums are moving out for every 2 potassiums that are coming in. So we end up with a lot more sodium outside of our cells than in, and a lot more potassium in than out. ATP energy is used to change the shape or conformation of the protein, and the affinity or the attraction of the carrier protein for either sodium or potassium changes when the shape changes. So these sodium-potassium exchange pumps are found in almost every single cell membrane, and there's hundreds and thousands of these sodium-potassium exchange pumps in just a single cell membrane. And this establishes this status where there's lots of sodium outside of our cells and very little in, and lots of potassium inside of our cells and very little out. And this is going to become important for the function of the cell membrane called transmembrane potential. So students have a really hard time remembering where's the sodium and where's the potassium for the most part. And so hopefully this little picture will help you to remember that. This crazy thing that sort of looks like a purple tree sitting in the bathtub, that's supposed to be a type of cell called a neuron. And if you notice the Ks, the potassiums are all inside the neuron, whereas the sodium is outside the neuron, bathing the outside of it. Hopefully that will help you to remember that the sodium is outside the cell and potassium's inside the cell. The last type of transport is bulk transport, also known as vesicular transport, because it uses things called vesicles. Vesicles are little small membrane-bound bubble-like structures inside the cell. And there's hundreds of these vesicles inside of our cells. With bulk transport, what we're doing is we're moving things both into and out of the cell in bulk. If we're moving things into the cell, that's called endocytosis. And there's three types of endocytosis: receptor-mediated endocytosis, pinocytosis, and phagocytosis. And then the opposite of endocytosis is exocytosis. So with exocytosis, we're doing bulk transport out of the cell. So let's take a look at the three types of endocytosis. In the top panel here, what we see is the cell membrane is pinching around a bacterial cell. You notice that here, the cell membrane is almost completely pinched around the bacterial cell. Once this part of the cell membrane meets that part of the cell membrane, we get this, a vesicle inside the cell. We call this particular vesicle a phagosome. This process here is called phagocytosis. Phagocytosis is sort of like a cell eating in bulk. Phago means to eat. So we're bringing in larger particles into the cell. And this might be for nutrient purposes, or it could be for like immune system purposes too. In this picture here, we see pinocytosis. Pinocytosis is the equivalent of a cell drinking. Notice that the same process is happening where the cell membrane is pinching around something and bringing it into the cell. But in this case, we're not bringing in large particles. We're bringing in mostly water with small solutes, hence the cell drinking. So that means the bottom picture is receptor-mediated endocytosis. The purple, I know my colors, I swear. The blue sort of Y-shaped things are receptor proteins, and upon binding to their target molecule, the process of endocytosis will occur. This is how things like, maybe you've heard of LDL cholesterol. This is how like LDL cholesterol gets into our cells. But I'm not going to spend a lot of time talking about that particular form of endocytosis. So this is a picture of exocytosis, and it pretty much looks just like what I showed you for endocytosis, but in reverse. Here we have a vesicle inside the cell that travels to the cell membrane. And when the vesicle membrane meets the cell membrane, the two fuse together, and then the contents of the vesicle are released outside the cell. So this is how neurotransmitters are released from neurons. This is how some hormones are released. This is how we get rid of some wastes from the cell. This is how we can release some things into our sweat, for example.